He has developed extensive tools to analyze genetic and genomic data, and he's recently participated in developing the single cell web portal Atlas and Workbench, and for exploring COVID-19 immune host responses. And with that, let's make sure you can share your screen, and then we'll turn the time over to you, Dr. Aronow.
Thank you, Diane. Yeah, I'm excited to be part of the CEMH. And yeah, I've just always loved blood cells and the immune system. And it's maybe not as much as others, but Irv Weissman was one of my favorite professors as an undergrad. And just between Irv and Hugh McDevitt, just learning about the architecture of the immune system, I think has been, it was always an inspiration to me. Yeah, and then recently when I got COVID last year, and one of my main long haul effects was developing ITP. And so I've been teetering around like 3,000 platelets at various times over the last 2 years. So that was kind of an exciting way to get into platelet megakaryocyte development biology in an interesting way. Actually, the drug that I'm taking, it's amitromypin, Doptelet, absolutely spectacular. Just one of these pills once every 4 days, and my platelets are fine as long as nothing's going on. So it's very paradoxical because it's a this is a consumptive autoimmune process, but here you're boosting platelet development generation by the megakaryocytes. It's a TPO analog. So yeah, a lot of hemodevelopmental biology in the flesh. Okay, so I'm going to tell you about this tool that we have that, am I not sharing my screen yet?
Your screen is not shared yet.
Oh, sorry.
There we go. Perfect. Thank you.
All right. So here's an intro to ToppCell. And I don't want to spend too much time on PowerPoint version. I want to go into the website and actually get you to think about how to use it for stuff that you care about. So this has been a project in evolution ever since we got single cell data, realized what it could do and how much biology it could tell us. I've just been extremely excited about modularizing the results from single cell data and then putting them into your fingers and then letting you arrange Ouija boards to help you understand where it's coming from and what it's doing. And so we're part of a couple of different consortia that have a lot of single cell genomic projects going on in them. Lung map, brain map, then IMH, Ped Cell Atlas as part of the Human Cell Atlas consortium, Digestive Health Center, Digestome, and then of course we had to get into COVID as deep as we could, and yeah, a lot of different efforts and a lot of cool people. This Kang Jin is current superstar graduate student who's really pushed the envelope on a lot of what goes into ToppCell. Eric Bartis is the principal engineer behind it, and has done some absolutely amazing things to make it really dance and sing according to various compositions that you want it to read out. And it was actually started by a graduate student, Yunguan Wang, and other just great people to work with here as part of it. And ToppCell is part of a suite of this TopGene suite that lets you link up a lot of information about genes and their functions and behaviors. And so, okay, so here's the general goal of ToppCell. It's to make single cell data go into this world of knowledge and observations, and where you can really string things together and see what is it telling us. And so the approach is to dissect tissues through their cell compositions as tissues go through different biological states, developing, undergoing disease process, infections, response in time courses, all sorts of perturbations, and trying to infer what's the emergent biology that comes out of the tissue when it's functioning properly and responding to perturbations. So we want to use all the single cell data and everything that we know about genes, everything we know about the cells, and somehow string it together and use it to navigate through this data and to learn. So how do we do that? Think about a system for a second. System is a tissue has inputs, it has outputs, it has properties. It's a collection of entities, the cell types. They're in various relationships to each other. They can have basement membranes, they have direct connections, they could have homocellular interactions, heterocellular interactions, and so it's all through that there's emergent biology that comes out of it, and so we know so much, vast amounts, more than even the any one expert in any one pathway, any one cell type, any one organ knows. And so computationally, we should be able to put this stuff together and really give you almost an augmented reality of the way that you're looking at data and trying to understand your system. So here's a view of how molecular entities form cellular entities that gather functions, and these are the kinds of functions that we know about cells and genes, and then out of the tissues comes things like physiological function or abnormalities of physiological function that we'd call disease phenotypes, and it's all through this structure function interactome. So this is the kind of data that you want to use when you analyze your single cell data. So look at it from the perspective of a single gene, and then think about the whole genome. From just one gene in the genome, there's all sorts of information that is part of the gene or that comes out of the gene based on what it encodes and what those coding products, encoded products do, and so there's all about its regulation, where it is in the chromosome, and regulation of its mutation, response, transcriptional control mechanisms that are either part of normal signaling networks or perturbation networks. And then the proteins that come out interact, and so we use those protein interactions and biological processes. These are all kind of themes of knowledge that link up different genes. And so when we look at a single cell type and think about other types that are in relationship to it in a tissue, we want to use all of this information to kind of better understand that cell network that's coming out from this juxtaposition of heterogeneous cells within a tissue. So I think that's a level even that's beyond understanding the single cells themselves. Emergent properties of the system, and we want all of these single cell or the gene expression signatures of cells to help us understand how all of this works together to make complex function out of organs and cell types. And so the end goal may be just to produce the right amount of platelets that have the right properties to do hemostasis or any other cell type and its functions. But in order to do that, there's a lot of dynamics in the tissue environment, usually bone marrow, but other places too sometimes, where the cells are produced and their levels are controlled, their release is controlled, everything. Okay, so how do we string that together? Here's a lot of where single cell data is at right now, truthfully. We're still involved in trying to cluster out data, get it properly normalized, get, really understand what these or highly similar cell types are within these different clouds, and if we make if we can adjust for variations in the this batches or origins of the sample or other sampling depths that vary within the cell type, just because it's sort of inherent in the technology. You can come up with these very crispy maps of cells that are close to each other, very far from each other, seem to be possibly highly similar to each other, but with a shift. But to really understand what are the properties of each of these states, we've got to represent those states in an organized way, and then figure out how they relate to each other. So let's say this clusters 10, 2, 8, and whatever that is in there, 7, are a part of a lineage. Is there a set of genes that are part of that lineage that are very important? Let's say they're myeloid cells. And so for all myeloid cells, or all T lymphocytes, or all epithelial cells, are there signatures of genes that sort of span those lineages or classes within which there's subclass structure, and so the genes of each subclass then work in that framework of the class genes to then have thematic function and then dedicated function that's specific to one of those states. And so following that logic, that gives you really a different approach to how to analyze the gene expression signatures that are present in a data set like that. And so that's what we do in ToppCell, is we basically try and find cellular hierarchies, class, subclass structures, lineage, class, subclass, and cell type, and then maybe cell type substate, that drive the finding of these different gene expression signatures that can span different levels of lineage or cell class or cell type, that sort of thing. And so then when you think about any one of these classes in here of cells, types of cells, it can be the result of really a compounded signature of genes which are part of that whole. I don't know if can you see my cursor okay? Then this
Yes,
module,
we can. Sorry.
plus this module, plus that module, plus that module equals the genomic expression of that cell type in here. And then you can use that to figure out, okay, by each one of those modules, what are the genes and pathways that are coming out as a result of the selection of those genes as part of the the ensemble of that state. So that's the thinking, and how do we kind of materialize that and put that in your hands so that you can work with it. So so that's what Eric and Kang have been up to, and developing. There's a couple of things that have to happen after that clustering and annotation that then put these gene signatures into a state where you can now look at them, compare them to each other, and then operate on them, and then figure out what's the biology that underlies it. And so the goal of ToppCell, and this is kind of what I want you to get out of this presentation. I want 6 main points. For a given gene of interest that you think is really important in your system, how is it expressed across a series of these cell types? So you want to do a gene level query into multiple data sets, and then get some sort of a view out of what's that gene doing. And then you can decide, oh, I'm going to study it. I see that, let's say, for example, it's expressed in 3 cell types that are kind of connected within a lineage. And maybe each of those subtypes, the gene will vary in its expression. Well, is the gene doing the same thing in those 3 cell types? Does it have a major role in just one of them? In other words, are the states of these cells, the differentiation states, shifting in such a way that now the function of that gene is a little bit different. It's working in a gene network in a little bit different way. So you can now use those 3 states and then to look at what are the other pathways that are implicated by its interaction with other genes of each of those states. So that's really a study of the overexpressed genes within those cell types. And then if we can make this library of all these signatures from all these data sets, can you now put in a signature into a database, do a query, and then see how does that signature from your cells that you're studying, how does that compare over a very large database to other signatures in those cells? And then, this is the question that I've really been fumbling around with in this whole intro is how are you improving your understanding of each cell type as a result of looking at these gene modules and their properties, inherent properties in the genes that are part of those signatures. And then once you do that for one cell, then you can do that for a series of cells, and then you can kind of better understand how a series of cells differ in their genomic biology, their pathway biology. What is it that drives them to adopt a different specialized set of functions? And then for all of that, and all of that prior knowledge, and all that you're calling up when you do these pathway analyses and enrichments and gene networks, and then you go back and say, okay, for all of that, how many genes of a cell's signature am I really connect? And the answer is usually maybe only about half of the genes of your signature can you really account for. And so what about this other half of the other genes that you don't have great information about? Can you look at some of their properties of those genes and then kind of prioritize the likelihood that some of those are working in the exact same pathways that the other genes are working in? So anyway, so that's the agenda. Let's see how we do and get into it. And yeah, so let's start off on the website and see what we can see. Any questions from that point of view?
So far, we don't have any in the chat, but anyone who has a question is free to ask.
And I just realized that I'm getting incredibly improved performance of the system using Safari on a Mac compared to Chrome. So I just all of a sudden switched over to Safari, and just to make it difficult on myself to navigate the interface. But okay, so here's ToppCell, just toppcell.cchmc.org, and then here's the way that we've got projects organized. A lot of this organization was Kang and Eric just working amazingly to do this. And so we might shift the name on this, but to blood cell map. But right now we're in immune cell map is where we're putting a lot of the hematological data sets, and really just starting in that. But there's other places, like just on general mouse atlas or human body atlas, body map, other places where, and even in the COVID-19 atlas, where there's going to be huge amounts of hematologic cell types that are doing really interesting things. So you can either go focus in on one of these areas, or you can do searches against all of the data in the entire system. But I'm tempted to do a search here. Yeah, I'll do a quick one. Okay, so here, let's try AVP, one of the top genes of hematopoietic stem cells. oops, stem was corrected inappropriately. Oh, so this is, how in the hell? AVP. So you put in the gene, and there's a quick resolver to find the official gene symbol, and then you do a search with it. So was that big enough to see on that interface? I'm on
We
my
can
little
see it.
I'm on my little monitor so that I wasn't like assuming that you'd be able to see.
We can see it.
Okay, so here's AVP, and I put it in as a lowercase, and it looks for both the mouse and human ortholog, or it'll even find the ensemble entity that's been mapped to it. If that might appear down here later. And so what this is, I did it against all data sets in the whole ToppCell database, and and what you get out is, okay, here's what you queried. This is a project where it appears. The data set where a given signature has been pulled out. Here's the the data sets. Sometimes we shred them out to modularize them, get gene lists and get differentially expressed genes by a couple of different approaches. So there'll be a job name that's associated with it, and then from that job name, there'll be a given module that contains that gene, and this tells you the rank of the gene within the module, what its mean expression was in the log to TPM value, and then what its statistical score was for the specificity of its expression in this test, which is specified right here, which is polydendrocytes versus all cells of the substantia nigra. And so let's say we go down here to CD34 positive hematopoietic stem cells as compared to the world of human bone marrow atlas, or just against progenitors. So that's pretty good. That's what I was hoping for. I didn't know that would come out.
How is that helping us? Are you going to show us that in the context of hematopoiesis?
Yes. So now, there's two. You have two options here. That sounded a lot.
Well, it's published.
Diane.
It's expressed in that population.
Yeah, so you can, now here's the link that lets you go to that data set. And so you could just pop this out and go and see the whole data set. This is what happens when you do that. And then this is like a preview version of a data set. This is an old data set, bone marrow atlas from the Broad. And I need to get rid of this so I can see. And then here's how it's divided up. And so this was, you have to remember that, I was looking in the progenitors. Here's the signature. Here are the signatures of the progenitors. And remember, it was in the CD34 positive cells, HSC cells, when they were compared to the world. So if I just pop up this gene list from this view, it then shows me the list of genes in there. I'm going to just find.
What's this list of genes? Genes that are co-expressed the same as AVP?
Yes. So I remember we searched for AVP. It found that data set. It said that it was in CD34 positive HSCs when they were compared to the world. In this case, it was, oh boy, we misspelled hematopoietic. In this case, it's saying that the gene was the number 7 or 8 gene, but it's interesting that that when you looked back in the results, here from modules that contain AVP, it said it was the number 1 gene of that module, and this link over here lets you pop up that heat map. Let's just do that. So I click that, and now this gives you a very concise way of showing the gene expression signature and the cell types that are present there. So this is just the average. In this case, this was 370,000 cells that were averaged down into here just 36 different cell types. I'm right-clicking over here and then I'm going to say Show Text, and now that shows the labels of these cell types, and then here I can do the same thing, right-click into the lineage, Show Text, and so now here's this class of cells, the progenitors, hematopoietic progenitors, and then these are the subtypes of cells within there. And this is actually showing a defect here of, here's hematopoietic progenitors versus the world, and this was a comparison of the CD34 HSC versus just the guys that I've highlighted there, those progenitors. Sometimes that's not the most useful stat test, but sometimes it does find some other genes. So here I'm going to just put in AVP into this interface, and now here's where AVP is in this list. And there's a couple of different pieces of information about it that's shown there. I'm going to say show this in the tooltip, and I'm going to control this and say give me, let's expand the display so I'm showing 20 pixels per row. And then now I can just cruise down here and see, okay, where's AVP? There's AVP, and you can see, oh wow, that's why it really scored #1 in the comparison of the HSC versus the other progenitor cell types, because it's really incredibly exclusive to the HSC amongst the progenitors. And so this is just, look at that. That's a hematopoietic progenitors versus the world, which is the class level comparison. So this was for looking for genes that are just sort of expressed broadly in this group compared to non-hematopoietic progenitors. So this is just a starting list of genes that you can work from. So let's back out of here and then try and get some orientation. So here's this HSC signature versus the world, and there's a problem here that there's all the ribosomal genes come up as scoring really high in expression but not necessarily in significance. RPS, RPL, and so now I've just highlighted the ribosomal genes, and I'm just going to right click over here and say invert the selection, and now I'm going to just get rid of the ribosomal genes, and there's a little button here that says make a new map without that the ribosomal genes in there. Okay, so now you've got two signatures that are kind of hovering around. This is just progenitors versus the world, and this is the CD34 specific genes. And notice there's a score over here of genes that are this is the statistical test, the score. In this case, it's the T score for whatever this test is. In this case, it's 34 versus progenitors, or progenitors versus the world. And so this is a great signature for progenitors versus the world, and the significance of the genes is really high. There's not that much that's super specific to just the CD34 HSC. And the reason why is because a lot of these genes that are highly expressed in the 34 cells versus progenitors tend to be they can be a little bit more broadly expressed, and the same for these genes into other lineages. So there's a boost of a bunch of genes in the stem cells compared to the non-stem cell progenitors. And then there's also this phenomenon of shared expression across all the progenitors. So how that pans out, and what does that mean, and so and what are the properties of the HSCs themselves as compared to other progenitor cells? That's where we can help you with the next steps after that, and it's really cool to do that. Let's just do a quick sanity check of what these genes are and what they're connected to. So I just highlighted. Let's see all the genes. I'm going to just say select all, and then I right click into this column of human symbol and go to top gene. So top gene reposits all this information, including the cell atlas. So this is a top fun analysis, just a functional enrichment analysis. And somehow I did not get the right. Let's copy. That's interesting. It's or not, depending on your point of view. I've never seen that. That is an extremely odd defect. I wonder if it has anything to do with between two browsers. That would make 0 sense.
So while you're trying to get that to work, Bruce, I just want to ask a couple of questions for the audience and myself. You're saying that's where we can help you. Do you guys have like times we sign up to try and come online? And I mean, I'm trying to figure out
Ooh, that would be cool.
how we get help from you.
Just email me, and I can do this in a directed way with you anytime.
Perfect.
I have no idea what is the problem. It's something related to clipboard, memory, Safari. I am, boy, using Safari probably was a really bad idea. I'm going to do this one more time. I am going to switch to
for those of you who are interested, I just put Dr. Aronow's email address in the chat so that you can reach out to him for your favorite gene.
All right. All right. So this is that essentially another data set, or maybe I can't remember. Okay, it's the same data set, and now we're just showing the best signatures of each one of those cell types. And so this is B cells, erythrocytic, erythropoietic. Here's the progenitors again, and then megakaryocytes and myeloid and lymphocytes. And so here's the HSC set, and where's the HSC genes? Right there. Yeah. Okay, so we've got all of these genes, and this is a little bit when you put it all together, this is what I was implying, that the stem cells genes are not necessarily super specific. They're shared with a lot of the progenitors, and then what's happening is that in the other progenitors, they'll have that core signature of genes, and then they grow additional genes on top of that. And there's some degree of shutting down of that core signature, so it's that they're running on this core signature, and then as they differentiate out towards erythrocytic or myelocytic or platelets to make megakaryocytes, they'll add in new modules and then take away a few from here. But anyway, here's the core signature of the HSC, and we'll just go and compare it. So let's say I've gone through this analysis. It's a new data set, and I'm going to compare my signature that I get from here. And this is just the HSCs in this one data set. I'm going to compare that to all the signatures in the database. And so I popped it in here. And when you submit to TopGene, you get control over, let's see, make sure that everything's found. You get control of what it's going to be compared to, and this is this part of TopGene that we've been building lately, the ToppCell Atlas. And if you open it up, it lists all the different data sets that you can compare to, and so you could do all of them, or you could down select to different ones. Let's just do all of them for the time being, and it should take care of trying to find the most specific overlaps to that core signature. Okay, so here's some interesting properties of that gene list, and some of it is a little bit specific to that data set. It's not all something, these aren't, it's not a, you'll see down in the signatures, it's not a perfect hit to other people's stem cell signatures. But great enrichments for hematopoietic precursor cell abnormalities and hemopoietic genes. If you just click on this link here, then you can see what the genes are that overlap with that category of information. So that GATA1, GATA2, BCL11A, fabulous, all great stuff. And now quite an overlap to hemostasis, which is very interesting. It's like they're poised for megakaryocytic biology. And then cis elements that are enriched. Okay, so it's down here in, these are old gene expression signatures before single cell data. There's all these signatures of genes that were derived from comparisons of bulk RNA-seq, maybe in FACS purified or genetically altered. These were FACS purified cells by Patrick Pattison in our consortium of the PCBC. And here you've got the best overlap by just one gene, 54 of them against his the HSC signature that Patrick could pull out. And then here's the overlap of those signatures, and this is it's a slightly different version of the bone marrow atlas, I think, than what I just pulled out. But it is from that data set, and so now you can see, okay, how do I actually compare to these different differentially expressed gene lists that came from this very data set? And this is a very cool thing to see. So now you get the biggest hit is to actually to MEPs and then HSCs, and then it trails off to lower and lower numbers as you go out these different NEPs, and depending on what the comparison is. In this case, it's just MEPs to the hematopoietic progenitors, or here versus the world, the CD34 proerythroblasts to the world. So it could be troubling to you in a lot of different ways that it's not a better hit to just pure HSCs. And when you look at sometimes the HSC signature itself is actually better represented in cells like the NPPs or the MEPs. Don't despair. This is a very specific question of the core genes of the HSC that are the most strongly expressed, that are specific to it. Sure, those genes actually, a lot of them turn on to a higher level, and when it goes out to an MEP, but the key is in the MEP or the MPP, an additional module comes on in addition to that core HSC signature that wasn't there in the HSC state. So now again, it's you go from this core signature, which is a totally fine signature that works across multiple subclasses of the differentiated state in early hematopoiesis, but it's that the acquisition of this extra module that now is what's more determining of the biology of the MPP or the MEP to get there, and that really is kind of a really good illustration of why we're taking this approach of hierarchical modules that should be used in your consideration if you're thinking about the genomic biology and determination of what those cells are. Does that make sense? It's a funny argument. I want to there's another couple of issues. Here's this new data set that we've is just now available in ToppCell, thanks to Eric working late last night, and it's a compendium of immune and hematopoietic cell types that are what in the hell is that? How does that happen? All right, somebody did that, and how do I clear that? How do I? I'm stuck.
I wonder if you can unshare and then reshare.
Good idea.
Part of our challenge is we try and make the Zoom, there we go, available more publicly, and that's the first time we got bombed, I think. There it is again. Sorry.
From David James.
Okay. Yep, I don't know what to do about that. One more time. Try it one more time.
Okay. Can you change the preferences?
That's a good question.
Any thoughts? Would anybody like to? So I was actually giving you a little bit of philosophy on how I look at differentiated states. There's something I wanted to show you that relates to the genomic biology of those, what's going on. And let's see, how can I say that better? I don't have it handy.
I'm still trying to figure out, Bruce, how to fix that issue. If you want to try and reshare again,
Okay.
we'll see if that's cleared up. I don't show a David James in our group, so I don't know what that means.
Yeah, well, and I'm seeing my name attached to a second login, but I know I'm only logged in once. Maybe someone's trying to,
So delete that name. Delete that second one that doesn't have Eric's picture.
I am the real Eric.
You are the real Eric.
Actually,
Yeah.
unless Charles Xavier is real, I would delete that one too.
Okay.
All right. So this is an extremely cool data set here that lets us compare hematopoietic cells in multiple tissues. So there's actually 24 tissues that blood cells appear in, and so it lets you compare these lineages of that are present in visceral tissues versus developing tissues versus maturing tissues, and it's a very neat thing. This isn't a good way to do it. So, and go to blood atlas, immune map, and then down here, it's called this immune hematologic cells from 24 different tissues from the tabulus sapiens. So this is a CZ Chan Zuckerberg initiative project, and they basically have, there's two, they use both 10x 3' v3 and SmartSeq 2, and then there's 24 tissues, and I put everything that's not thymus, spleen, lymph node, bone marrow, and circulating blood into tissue resident organs. And so now we can do a comparison of, let's say you just look at, let's look at macrophages in the different locations. So now you can go pull up macrophages from the lymph node versus bone marrow versus blood, and then versus spleen and thymus. Let's do sorry, this is select macrophage, and then even within that, you can open this up and then sometimes get some very interesting subtypes. Okay, now here's the big question: is and then comparison to other tissue resident macrophages.
Hey, Bruce,
And-
can I interrupt and provide a little context to some of what's on the screen there? So I don't know if you can see at the top of each group, there's a purple bar, and then there's
Hold on one second.
Mhm.
Go ahead. Okay, there you go.
and then there's a light blue bar. And so what the algorithm is doing as it's generating this hierarchy of different assays and tissue groups and lineages is it's going over every gene in the master expression matrix and finding within the context of both the light blue and the purple, what are the best expressed genes within that group. And then the groups are selected from, which cells are part of, say, myeloid dendritic. And then, so.
Here's the tissue resident tissue group, and then here's the mesenchymal cells of it, and the mesenchymal cells are getting compared to tissue resident. And then this is, Eric's talking about this purple bar here that encompasses tissue resident cell types, samples that were then that had all their cell types in them. These were the mesenchymal cells of the tissue resident, all the cells of the tissue resident organs that were pooled together, and the comparison was just that little dot in the middle to that purple group there, and that's what gave this, in this case, the mesenchymal cell type signature there that you're looking at. And there's a couple of different controls in here. You can download these different different files. This C-type file is this is a list of all the single cells in the entire data set. And then I don't count how many they're there, but it's like 170,000 cells. And then for each one of them, there's a designation about which technology was it from, what tissue was it from, and then what's its annotation of what cluster, Louvain or Leiden cluster did it have, and then what was the annotation of it with respect to class, subclass, and everything else. And it could be from a given individual. In this case, I don't remember how many individuals there were behind these this database, but now here is I requested the macrophage signature from the tissue resident. Here it's macrophage signature from spleen and blood and lymph node and bone marrow. So let's put it together. Okay, so how do we do that? I'm going to first sort by lineage. I clicked on this little button here, lineage, and then I'm holding down the shift key so that it holds that sort order, and then I'm going to sort by cell group, and then I'm going to hold it down one more time and go cell type. And now here's all the macrophages right there, and it's all part of the myeloid signature. And you can see a lot of those genes actually are expressed pretty well in other myeloid cells, but let's see, this is in my way.
Yeah.
So okay, so now the situation is that we've got really very different signatures in these different macrophage subtypes, and then the problem is, how do I sort that out and get something, find out what's the what are the extra genes that are there in these visceral tissues that say are not present in spleen, that would give splenic macrophages properties that might differ from other locations. And there's a lot of work to be done to figure that out, but and it's actually pretty easy to do in some ways. Let me show you one more button that would make that possible, but I don't know. Diane, are you still on?
Yes,
I
why?
am.
Oh, Diane
Diane
Krause.
Ward?
I was
Diane
thinking
Krause is
Krause.
still on, and so is Diane Ward.
And what would make the most sense here in terms of so that the hypothesis would be, oh, there's location-specific functions of macrophages that maybe could clue into special jobs that they're doing, either to, let's say, help pull in cells of or help deal with cells that need to be phagocytosed or deal with development of cell lineages. So I think the theory is that you could get clues about that by a comparative network analysis of the signatures from each one of these macrophages. So and there's an actually amazing approach to do that. So I've got those genes, those same signatures selected here, and now I'm going to go and hit this button over here on the far left called enrich, and so then that sends those 4 gene list signatures to a feature analyzer, and we're going to do a functional enrichment analysis of them and just look for just a couple. I'm going to basically just look for something different in their biological process or the effects of knocking them out. And so I'm just going to look for like the top. I'm going to do a network generator, and I'm just going to look for like the top 25 things about these 4 signatures. Okay, so then go down here and you hit this button, and then you get back a result of the connections of each of those signatures to different functions. And so it turns out they're quite similar. Here's the first time that they're not is in this. So the things that it's pulling over: myeloid cell activation involved in immune response, neutrophil activation, myeloid activation.
Are you asking it to tell you things that are similar or things that are different?
I'm actually, there's both. I mean, there should be a lot of similarities. They're all macrophage. There are macrophages in those 4 different locations, but is there something about them that's actually specific? And there's something about activation that comes out as being different. I'm just going to zoom out a little bit, and so now as you get down to lower ranks, then now you're going to see stuff that's very specific to just one of those macrophage subtypes. So this was the blood macrophages, monocyte derived. Let's look at the bone marrow macrophages. And so just see if there's something that comes up that's specific to the bone marrow. And yeah, here's one: innate immune response. So it's these genes here with C1q, B and A and CARD9, CD14, CSF1R. So there's something about this innate immune response which is different in the bone marrow than spleen. You'd think that would be like spleen and lymph node. It would be very important, but apparently not quite. And then you go down to, this is mouse knockouts. Again, everything looks mostly the same, but then you get down in here and then you see stuff that's something about interleukin secretion. So a bunch of genes that are involved in the control of interleukin secretion are different in the context of bone marrow versus most of the other tissues. This is, I think, somatic tissues over there. But and inflammation, anti-inflammation, coagulation is higher in one of the other cell types. Humoral immune response, something about that role. Tolerance, so and that's coming out of the marrow. So the implication here is that, hey, the macrophages are doing something that is relevant to regulate tolerance formation, and so that it may actually be interacting with B cells or T cells in the marrow environment. And you can actually explore that too by going here and then just selecting. I'm going to just select B cells and T cells. That's from the lymph node. I'm going to just do the B cell T cell signature for marrow, and to do that, I have to open marrow, open lymphocytes. Here's the B cells, here's the T cells, and then do that same question. I could limit it to ligands and receptors. It might be kind of more focused to do that. Kind of fun. And now send that over to that same system, but we're going to now look for interactions between the cells. So this button stays selected there for an interaction network, and then hit run, and then it looks for all the protein-protein interactions between ligands and receptors between the macrophages and the T cells and B cells. And here's B lymphocytes and T lymphocytes, and it takes a little work to pull it apart, but basically you see these connections between the different cell types, and then it's really you have to download this and then open it up in Cytoscape, and then pull it apart and see where is there anything here. And then there's stuff like CXCR5 and CD40 ligand and IL receptors and interleukin CCLs. This really has to be teased apart. And why exactly the bone marrow macrophages here's their connections to the to B cells, and there's TNS SF13 is involved in there, LTBR, and so that really, I mean, those are already some clues that CD40 FC receptor, that where there's something a little bit different about those macrophages in the bone marrow context that maybe is helping eliminate self-reactive clones. I don't know. So that's kind of actually have no clue whether that makes any sense. But those guys there's B cells under maturation and T cells under maturation. There's way more T cells in the bone marrow environment than I thought, and that's actually another angle of attack where you can really display the relative numbers of each of these cells per these compartments. And I was actually surprised to see, just when you look at different bone marrow atlases, that maturing B cells you would expect, but maturing T cells in the bone marrow too, I don't know is that a thing? That there's a lot of them. Proportionally, there's more maturing T cells in the bone marrow than there are than B cells even.
Since there isn't a large immunity population on this meeting, I don't know, but I'd have thought it was in the thymus.
Yeah, but it's a different stage. Yeah.
Yeah.
But even but just this, I guess the bottom line of all this discussion is that this provides you with a foundation for then a comparative analysis across different tissues and what's going on with cell activation and maturation in these different compartments. And this is still cutting edge stuff. This is still beyond what our state of knowledge is really in terms of understanding the cell dynamics in these different tissue contexts. And I mean, we know that that's a thing and that goes on, but in terms of having real control over the data, so that the data is able to talk to us out in these different directions, and we can be well informed by comparative analyses of these cell types in the different compartments, and make sure that holds up in different studies using nuke-seq, using cell-seq, using site-seq, all of those different approaches. Are we, is this a robust finding? Is it reproduced? Is it, sometimes some technologies show some things that other technologies are not able to show us. But
so Bruce, I'm going to just interrupt 1 second just to keep us kind of on time. People have been here for an hour. I want to see if you have a summary slide or something, or if people want to ask questions before we stop the presentation.
Yeah, I guess given all of that, you can see how you can go out these different directions of, finding cells of interest that a gene of interest is on in, how you start to look at the genes within that signature, and how you have to compose multiple modules together to get a better understanding of that cell type, and then a comparison of the subtypes within it. All of that stuff, the problem of understanding hematopoietic stem cells is, really brings out all of that in spades. That a lot of times it's just not only about the positive overexpressed genes of that cell type. A lot of times it's you have to understand what's not there in that cell type compared to other cell types that it's adopting a separate phenotype from. And so, yeah, so different angles of attack. There's so many angles of attack, and there's so many ways to bring in prior knowledge to help you, understand these gene networks and what are the emergent properties of the genes that are active in these cell types. So it's just so much fun. Anyway, that's my argument for why you should waste more time than you'd like to in going through this. But we're trying to make it easy for you to explore this, and certainly anything that anybody wants to do that they think is going to be interesting, I'm all about trying to help do that.
Great. Can I invite anybody to ask questions at this time? I don't see anything in the chat right now.
Yeah,
I've been
Diane, can I ask you a question?
Absolutely.
So it's really a nice integrated database. But if you look at the other database, like IMNGen and stuff, you just punch the gene in, and you get the distribution or cell-specific expression data of the certain genes. Now, the single-cell RNA-seq nowadays, it's what's big deal is the cell annotation. So if I have a list of the genes, so I identify 35 clusters, and then I have the DGE of each clusters, and then I have a top 10 genes, and then can I plug that in and then
Yes.
get can I?
Yeah, there's an approach that you can use where I'm not sure if I could generate it on the fly, but it's basically get it. You want to put your in column 1 the genes, column 2 the label of the cluster, and then and I could do it pretty quickly, but then you go to this this system top cluster, and there's this thing here called alternative entry methods, and use the tab separated so let's say MIB and AVP, and I'm going to go
So I'm going to interrupt for just 1 second, it sounds like it would be a good conversation for individuals to contact you and possibly even in a Zoom sort of step through this together to get familiarity with this amazing tool that you guys have developed for us to look at lineage. Is that an accurate statement?
Sure.
I've been trying to play with it over on my other screen on a gene I'm interested in, and I clearly can see that I need some like hands-on step-by-step help in it. So I don't know if that helps you at all, Aki.
Yeah, that helps. But I just have one quick, so to say, request. If you have a postdocs or graduate students who can come up with the R or Python script to access your database and do the cell annotation right on the R or Python, that would be great.
There's other, do you know about Azimuth?
Yes.
Azimuth is a model we really want to support. So we want to generate new Azimuth reference models that can be used directly, so you could just do it within Seurat.
Yeah, that would be perfect. Yeah.
Yeah.
You could do that. So how long is it going to take?
Satij is really, doing that extremely well, and
Okay.
the HubMap project is really putting a lot of that together. But they, they only have maybe a dozen reference maps right now. And what we're seeing is that you need a different reference if you're using NukeSeq. You need a different reference if you're looking at development versus adult, and
Right.
so and then how you recognize the outlier cells, and then adjudicate between for lower scoring cells that aren't getting mapped to the existing reference, what's the right workflow? That all that stuff is yet to be figured out. And you could argue that in this kind of looser compendium of cell approach that we support, that you can go between different data sets to then get the best annotations from many data sets that could.
Yeah, that'll be perfect if you can support a Surat or Scampi in the Python. Oh, that's great. So
Yeah.
okay, Diane, thanks.
Hey,
Yeah.
you're welcome.
Thank you,
Anybody
Aki.
else?
Yeah, I
Anybody
know
else
that in
wanna
our
ask?
data wrangling, we do use ScanPy and NumPy to do some of the expression matrix wrangling, especially when we want to start combining multiple data sets from multiple investigators.
Okay.
This new Surat 4 functions are really doing a lot of things right and pretty easily to give you a good starting point for that.
Yeah, load of bugs though. I mean,
Well, so here's a question that has to do with like these coordinating centers and you guys as a core. Is this something that people could apply for that type A funding to really get from the Centers of Excellence Coordinating Center to really get tutorials or what Aki's talking about, sort of program details that can connect better their pathway, their gene of interest and their pathway or expression in different tissues from the same kind of quote unquote cell type. Is that a thing?
It is a thing. I mean, it's not something that we're really kind of funded and staffed for. But if it's a cool thing, we definitely would make time to do it.
Okay, it's just a query because I have no idea, but maybe that's something, some communications between people who participated today might be interested in more conversation that might help with their
But
data
we
sets.
do have a Python package that does a lot of those functions of that Kang has developed, the Python package, and it does some significant parts of the early parts of ToppCell. That you could do that as a standalone function.
Okay, I'm gonna stop us there and stop the recording, but I want to thank everybody for their participation. And just ask if anybody else has like one final question that they want to ask. I don't see anything in the chat. I want to thank you again. I'm saying Bruce, even though we haven't met in person, this is kind of in person, but thank you, Dr. Aronow, for your presentation today. And want to encourage individuals who see that this might be a useful tool to reach out to Dr. Aronow and his group.
Use it.
Use it. Strengthen it.
And any suggestions or help that you need, please hit us up, and I'd be super happy.
Great. Thank you so much. And
Thank you, Diane.
everybody take care.
Bye.
Bye.